Really excited to introduce the next trio, actually. So why do we do all this work? We do all this work because fat is supposed to be tasty, right? But is it actually tasty? What actually is the point? And I think the work of the group that's going to be presenting next will start to address some of this. And I'm sorry, in the program, I think it just says Dr. Zhang Yuen, who's one of the presenters in this group, who's an entrepreneur in residence and the director of technology at the new Cellular Agriculture Commercialization Lab at Tufts, who does PhD in the Kaplan Lab at Tufts there. And he's joined by Emily Liu, who is a PhD student at Tufts as well, also in the Kaplan Lab, and also with Kevin Lin Zhang, who is a undergraduate student who's also joining and participating in this really exciting research. So thanks so much and welcome.
Oh, hello everyone. I'm John. This is Emily. This is Kevin. We're ready to give the best speech of the conference within the next 15 minutes, So last year we presented about our just making some pig fat, looking at the nutritional analysis, similar fatty acid ratios with the conventional or in vivo fat. And then this year we wanted to present about what it might taste like. So, oh, there we go. So again, as everyone said, we don't have to talk too much about the problems or negative externalities of animal agriculture, environmental degradation, all these reasons, also human health issues, zoonotic diseases, antimicrobial resistance, #1 issue of the World Health Organization. Essentially, meat production can contribute to the destruction of the world. But what about cultivated fat or meat as a solution? Perhaps it is slaughter-free, good for animal welfare, antibiotic-free, good for that antimicrobial resistance, pathogen-free for the zoonotic diseases, low land use, so maybe sustainability benefits, but most importantly, perhaps it is tasty or it smells good, because from the GFI report recently, for plant-based meats, taste was cited from surveys as the top reason for lack of consumer repurchase. So allegedly, supposedly, when consumers eat the plant-based meat, they think the taste is, okay, and then they don't buy more. So fat, we've already mentioned, helps with texture, species-specific flavors. So supposedly, if you take pork or bacon, remove the fat or lipids, then it just tastes like meat, but not bacon. And then intramuscular fat increases consumer acceptance, which is that thing that we're trying to improve. But does this apply to cultivated fat? When we started this work, there was very little data about whether the cultivated fat actually acts in the same way. Does it actually taste good? Everyone's saying, cultivated fat tastes good, but then we wanted to look into this more and have a more comprehensive look at this, whether they're the same. So our objectives, first, we wanted to generate some in vitro fat. So we used the method we made before, where we just grew it in the tissue culture flasks and scraped them. And we are also showing a little bit of cell line and media development from that. And then here's the main part. We wanted to, from the same sample, do a tandem quantitative analysis with the GCMS and the dynamic headspace sampling, as well as have some descriptive analyses with people actually smelling these cooked pork fats and describing it and stuff like that. And then the goal is to provide some data on whether cultivated fat, yes, no, does it actually help with flavor in cultivated meat or alternative proteins in general.
So to give everyone some context on the cells that we use for our study, we started with our pig dedifferentiated fat cells that we isolated a year ago, and we conducted a clonal isolation. So as you can see here, we screened over 80 clones for both proliferation speed and adipogenic capacity. So you can see here our top 15 clones, and then in the green are the 5 clones that we picked for further screening. So these 5 quickly, with further screening, turned into 3 clones. And then based off of their lipid accumulation capacity, we continued our screening with clone N47. And so we were able to track clone N47 through 100 cell doublings, as well as its doubling time, which averaged around high 30s to 40 hours. So we began with our clonal population doing some media optimization. And so you can see here our two-step differentiation regimen. We started with our lipid accumulation, we started with our induction media with IBMX. And so we were previously using a concentration of 0.5 millimolar, and we were able to actually reduce that to 0.1. We also optimized in our maintenance media for dexamethasone and rosiglitazone. So previously, we were using both in our accumulation media, and we found that we can actually omit the use of rosiglitazone and only use dexamethasone. So these are the efforts that we've made in our lab to try to reduce and eventually remove the use of these small molecule inducers. So to go over our workflow for creating the material we used for our study, you can see here that our clonal population becomes rather lipid-laden after 12 days of adipogenesis. And so we can then harvest them via cell scraping, and we are left with this aggregated fat tissue, which can be used for sensory analysis. So we started with our GCMS analysis. What you're seeing here are the total ion chromatograms of the cultivated pork fat seen in black, and then the conventional pork fat seen in red. And so these were extracted at cooking temperature, so 125 C. And we definitely see a lot of similarities and differences in their VOC profiles. Looking a bit closer, we actually extracted 130 to up to 175 volatiles from these fat samples. We only have space to show you the top 20 here. So out of all the detected compounds, you can see that the red ones highlighted are unique to each fat sample, and highlighted in blue are shared between the two. And so we have quite a few shared compounds between them, notably a lot of long and medium chain fatty acids. And so a big difference between the two is the proportion or the amount detected. And this data is still being worked through, so it's a bit preliminary. We are still trying to figure out what might be some of the best ways to analyze and present this data such that it's most helpful for everyone in the field. So we're even open to suggestions for anyone with expertise in this as well. So next, we wanted to know what real potential consumers thought about the smell of our cultivated fat tissue. So I was able to recruit around 55 students and staff from the Tufts School of Engineering to smell and give their thoughts on our cultivated pork fat compared to conventional. So this is a picture of me and Kevin with some of our sensory panelists looking very happy to be sniffing. Yes. So we first presented them with a discrimination test through a triangle test. And so our panelists were given 3 samples, 2 of which were alike and 1 of which was different. And they were tasked with discriminating between the aroma of cultivated and conventional pork fat. So you can see here that around 76% of our panelists were able to discriminate. So we determined that these potential consumers were able to discern between the aromas of cultivated and conventional pork fat when given next to each other. They were also given a descriptive task in the form of a check all that apply with a variety of meat-relevant aroma attributes. So most importantly, our panelists found that the aroma of the in vitro grown pork fat was fatty and savory and fried and bacon-like, a lot of attributes that would be attributed to conventional meat. So the exception were 2 of the more negative aroma attributes, musty and barnyard, which were more frequently used by the panelists to describe our cultivated fat. And so our panelists were also able to rank their overall liking of the aroma on a 7-point scale. And we found that despite some of these differences in the musty and barnyard smells, the preferences for either aroma seem to be statistically insignificant. But if you look a little bit closer at the data, we see that there might be a trend towards somewhat dislike for our cultivated fat and towards somewhat like for the conventional pork fat. And so we'd like to look into this a little bit further. So we also conducted a penalty, also called a lift analysis, with our aroma attributes, and this links the usage of certain attributes with the overall liking score that our panelists gave us. So we found what we expected, which was that some attributes that are associated typically with meat products, like bacon-like, savory, and roasted, led to an overall increase in the liking score, whereas attributes typically associated with rancid meat or lipid oxidation contributed to a decrease in this liking score.
So after we determined what the panelists smelled from the sensory evaluation, we needed to know why and what compounds were behind the complex, rich symphony of aromas that came from pig fat. So what we did was we performed an additional GCMS ODP analysis, ODP for olfactory detection port, which would help us associate the individual compounds that came out of the GC column with descriptive attributes for each compound related to the smell. And this would help us use this information to modulate the compounds that come out of the cultivated pig fat samples that we grow to hopefully make a better smelling, better tasting cultivated pig fat. And here you can, there's me. Unfortunately, I forgot to wear a suit to the lab that day. But there's me smelling outside of from the ODP port, and coming out are the different compounds and their aromas for each individual one. And this is an olfactogram of the conventional porcine adipocyte samples heated at 125 C. And you can see that unlike the total ion chromatogram shown in the previous slides, you can see the compounds are followed with descriptive words that are representative of the odors they produced. And with this data, we were able to build a flavor database where we can associate the compounds and their corresponding odors and categorize them with positive smelling compounds, like the smells that resembled meat, and the not so good smelling compounds. And you can see words such as cooked, oily, greasy, fat, and positive. And then in the negative, you can see like rancid, musty, sour, and etc.
So we're hoping that the start of this flavor database will be really helpful for us to begin to modulate the flavor of cultivated meat in the future. So in conclusion, it smells like bacon. It's fatty, savory, meaty, even bacon-like to some of the panelists. But further research is required to investigate what are the derivatives of some of these aroma-active compounds, especially the negative ones that our panelists detected, like the musty and the barnyard, so we can modify that in the future. We'd also like to investigate the development of these flavor compounds over time, such as through chilled aging, which typically affects the flavor of traditional meat as well.
And just for acknowledgements, thank you all for listening. We'd like to thank the David Kaplan Lab, where we did all this work, and Professor David, who gave us the mentorship and infrastructure. And then also the Maastricht conference, this is us back in 2019, as well as Dr. Scott Frost, our GCMS and sensory mentor, who's in the audience over there, as well as New Harvest for funding me as a grad student fellow, GFI for generally supporting the lab as well, and also our funding agencies from the US. Thank you.
Thanks so much, guys. All right, we have time for a couple questions. Yeah. I can also just hand you the mic, yeah.
There's an online question by Marianne James. She's asking, were any of the smelling panel non-pork eaters? And if yes, did they perform differently from those who eat pork?
So we did tell our panelists previously that they'd be smelling pork products. And so anyone who couldn't smell pork did not participate. But in terms of vegetarian and non-vegetarian, we did have a lot of vegetarian panelists, so people that don't engage in meat. And so we've definitely been thinking about that a lot since the panel and how that probably affected both their overall liking score and also some of the positive and negative attributes that they attributed.
Yeah, we can pass the cube over here.
Thank you. Very nice talk indeed. I was wondering, have you made any tests about the oxidation, lipid oxidation of in vitro versus in vivo pork fat that you tested? Was it more vulnerable or more resistant to rancidity, for example? And also, have you thought of, do you think it would be possible to maybe increase in some way the composition in certain fatty acids and these in vitro fats at some point in the future? Because this is one of the things that, for example, free-range meat is good for. Thank you.
Yeah, that's a great question. Thank you. And so we haven't yet looked into how storage would affect flavor. And so I think in our next steps, which I mentioned for this upcoming year, I would like to investigate age chilling or storage time and how that affects the flavor, because traditional meat is able to go through post-mortem age chilling, which induces lipid oxidation, which can induce some of those rancid flavors, but it also develops like the flavor that we interpret as meat. And so I'd like to investigate that in the cultivated product as well. And then I forgot your second question already, I'm sorry.
I think in our previous work, we've tried I guess the answer first is similar to what Johanna said. Applying the fatty acids to the cell culture can influence the fatty acid composition within the cells, but then probably not like you put in one and then you get one. As to short chain, we haven't looked in trying to give short chain, but then we're a bit wary about giving shorter chains because from the flavor database, we found that some of the saturated shorter chains or medium chains, like butanoic acid, like 8, 9, 10 carbon fatty acids were associated with the musty aroma and stuff like that. So I guess we never thought to put it in because of that.
Thanks so much. I also have a question, but I'll ask you guys later because I think we're out of time. So thank you so much. And it was very fun to have a trio discussion, right?